So, just a quick recap. What we were talking about in the previous lesson was the endomembrane system. Okay, the endomembrane system. Maybe I'll just do you don't necessarily have to copy this. You can just like watch, right? So we talk about how the rough ER, rough endoplasmic reticulum, is an extension of the nuclear envelope, and they are covered with ribosomes. So together, they produce proteins and package them, right? And so we have a vesicle, right? Plus protein. Now, does anybody remember where will these vesicles go? What's the next organelle that it goes to? That's okay. That's something to study for the next quiz. That will be the Golgi body. Okay. And the Golgi body will sort, modify, and repackage the proteins. We wrote it all down last time. I'm just trying to refresh your memory here. And once you repackage the protein, then it could go to different locations, right? So I'm not going to rewrite it, but you should review it on your own. It could leave the cell, for example. It can become part of the cell membrane. It could stay in the cytoplasm for some time until it is needed, or it could become something called lysosome, right? So all these we've written down last week. Okay? But I just want you to, recall how proteins are created. Of course, proteins does not have to be produced by the rough ER necessarily, right? They might be produced by free-floating ribosomes. Okay? Some ribosomes are just on their own, right? Floating around, and they too can produce protein. Okay. So now we are going to move on and talk about the rest of the cell. And there is this thing called peroxisome. Peroxisome. Now, here's a definition. Peroxisome, they are small round organelles that carry out oxidation reactions. Okay, so oxidation reactions, to keep it simple, they are reactions where electrons are removed. So these reactions where electrons are being removed, they tend to generate some byproducts. Okay. And the byproduct that is being created here is H2O2. H2O2, as you might remember, it is hydrogen peroxide. Okay. Hydrogen peroxide. Now, hydrogen peroxide is very toxic for the cell. That's why we can't have it, going all over the places. You need to contain these kind of reactions. Okay? And that's why we do them in the peroxisome. So we use the peroxisome to break down fatty acids and some amino acids. And it also helps detoxify poison. Now, if you remember, this sounds very familiar with the function for other organelles. Does anybody remember what other organelle is responsible for detoxifying toxin? Anyone at all? Yeah, the smooth ER, exactly, right? The smooth endoplasmic reticulum, right? And also, we talked about the lysosome being able to break down a lot of the macromolecules. So there are some overlapping functions here. In fact, the lysosome, which is this guy right here, looks very similar to the peroxisome. Like I wouldn't be able to tell if they don't label it, like which one is which, right? So the functions are also very similar, but the main distinction is this. The lysosome is part of the endomembrane system, okay, which is the stuff we drew last time, but the peroxisome is not. Also, the kind of reactions that happens are different. Lysosome uses enzyme, whereas peroxisome, we use oxidation reaction. Okay. So let me write down for you. Lysosome, okay, uses enzyme. I don't know if you can see that. I got to change the color. Okay. I'll just write here. Lysosome uses enzymes. They are also part of the endomembrane system. Okay. Whereas the peroxisomes, they do oxidation reaction. Okay. And they're not part of the endomembrane system. By the way, I should mention this. Even though quiz 2 is done online, the stuff that you are studying for, those will also be on the test number 1, right? Okay, so like, there is invested interest in you to remember those things earlier rather than later. All right, let's try this question here. In a moment, okay? Yeah, what's the answer? Yes. Anyone? I have one answer so far. That's good. You're good. All right. So if you track the movement of the protein produced from the rough ER, where is it going to end up? Well, if we look at the notes we took last time, again, the note is posted online, okay? So if you don't have it, you can download it and try to copy it. It can be released out of the cell. It can become part of the cell membrane. It can stay in the cytoplasm, or it can become a lysosome, right? All right. So if we look at all the choices here, it is not going to become part of the peroxisome. Okay. Peroxisome has nothing to do with the endomembrane system. Okay, next we are going to be talking about the mitochondria. The mitochondria, first of all, this is plural. Okay? The singular is mitochondria. Okay? Like, I mean, I don't really care whether you use the singular or plural. I just want to show you in case you read something and then it's spelled differently and you're wondering why, okay? So that's the singular, that's the plural. Now, this is the site of energy production. Does anybody remember what kind of fuel do we burn to produce a majority of the energy in the body? Yeah, louder, Josh, you have it right. Glucose, exactly, right? Okay, we take glucose and we're going to react it in a process called cellular respiration. So I'm going to write it out for you one more time. And it's about time to start to remember the reaction because that's something we'll be doing in lecture 5. Okay, so that's glucose, and we're going to be reacting it with 6 oxygen to produce 6 carbon dioxide and 6 water. And more importantly, it is going to produce energy. Part of this reaction occurs in the mitochondria. Now, depending on the kind of cells that we are looking at, some of them will have more mitochondria than others, depending on the function. What kind of cells do you think require a lot of energy to function? What is something in the body that's like always, working? That's keeping you alive. The heart, that's right. Okay, so if you look at the cells in the heart, right, you will have a lot of mitochondria in them, right? Because it never stops beating. The moment it stops beating, it's time to say goodbye. What about another organ that requires a lot of energy? Hopefully that organ is active right now, and the brain, that's right. Okay, the brain needs a lot of energy, right, to recall the answers on the quiz and whatnot, right? So they too have a lot of mitochondria. Right? Something like a skin cell. Skin cell don't do a lot of things. In fact, the surface skin cell, they're all dead anyways. But then the deeper ones, they don't really do much. So there's not a lot of mitochondria in them, right? Fat cells, they store energy for the most part. So there is not a lot of mitochondria in them. So it depends on the kind of cell that we're looking at. The number of mitochondria will be different. Now, I want to draw one together because understanding the structure of the mitochondria is key to understanding lecture 5. All right, so let's just try to get that sort out. Mitochondria structure. So they are oval shape, kind of like a jelly bean. And we call this the outer membrane. The outer membrane. Now, if there is an outer membrane, you can probably guess there must also be an inner membrane, right? But the inner membrane is going to fold out, right? So rather than being, like this, it folds up into these, usually they're called like finger-like projections. Right, so let's draw it out like that. It's gonna fold it up. Okay. They're like little stubby fingers, and it all fold up. Okay. So that is the inner membrane. It folds up into finger-like, that means they look like fingers, okay? Finger-like projections called cristae. Now remember, the mitochondria is where the cellular respiration, the chemical reaction that I wrote down for you, that's where it takes place. So whenever we have reactions, generally speaking, the more surface that's available for the reaction to take place, the more efficient, the faster you can do it. Okay? So by folding it up, you basically maximizes the amount of area available for reactions to take place. Okay. Like if you measure the entire area of the folded up inner membrane, that's a couple hundred times more than just the outer membrane. Okay. Between the outer membrane and the inner membrane, there is going to be a little gap, okay? An area, and we call this the intermembrane space. Intermembrane space. It's just the gap between the outer and the inner membrane. And last but not least, the very inside of the mitochondria, that is where you have the matrix. It's not like an empty space, okay? Like the matrix and the inner membrane space, they're filled with fluids. And in lecture 5, we will look at how these different structures play a role in creating energy for your cell. Okay. But for now, that is the mitochondria. Is that a question or just stretching your head? You have a question? No. Okay. Now, related to mitochondria is something called chloroplasts. Chloroplasts. Okay. Chloroplasts are not found in animal cells. Okay. Plant cells have chloroplasts. There are other organisms that might have chloroplasts, like the amoeba and the paramecium we talk about. Those are protists. Some protists have chloroplasts. Okay? But between animals and plant cells, only animal cells have chloroplasts. They are the site for photosynthesis. And I'll write the equation for you as well, but you don't have to memorize it. Okay? For photosynthesis, it is 6 carbon dioxide plus 6 water. And in the presence of light, it will convert these things into glucose and oxygen. Now, if you have been paying attention, you might notice that the chemical reaction for photosynthesis is actually reverse of cellular respiration. Okay? We wrote it down 2 slides ago. Right? Look at the cellular respiration. It is completely opposite to the photosynthesis. Right? So these 2 processes actually complement each other. Right? It forms some kind of balance in nature. So the green part of the plant, that's where most of the photosynthesis occur. And they're green because they have the chloroplasts, and the chloroplasts have chlorophyll in them. Okay, chlorophyll. So green vegetables, especially like really dark ones, dark green, like kale, that kind of stuff, spinach, they have a lot of chlorophyll in it. Right? And the chlorophyll is important because it allows the plant to capture the sunlight. All right. Some student in the past told me that they buy chlorophyll extract from online, I think, and they add it to their drinks because they believe it's healthy for them. So I actually looked it up and I found no creditable benefit of consuming chlorophyll. Okay, like if you want chlorophyll, you can just eat your vegetables. Like there's no need to take extra amount of it because we actually don't do anything with it. Okay, it's not like we can become photosynthetic or something. Now, the chloroplast is also double membrane, like our mitochondria. It has an outer membrane, inner membrane. You are not responsible for the structure of the chloroplast. Okay? That's all the information you have to know. I do want to show you this picture though. This is a nice diagram that summarizes the relationship between what's going on in the chloroplast and what's going on in the mitochondria. Right? Specifically, the ingredients, the reactants for cellular respiration are actually the products of photosynthesis. And so we take the glucose and we get the glucose by eating them, right? When you eat your starch, when you eat your rice, potato, and whatnot, that is how we get glucose. And then we combine it with the oxygen that we breathe, and we are able to breathe out carbon dioxide and also produce water. For the plant, they are able to take the carbon dioxide from the air, water from the rain, or like if it's your house plant, you water them, and then they will convert it into glucose. Okay? The plant then store the glucose as starch, which they themselves can use. Okay? So for the plants, they actually have both the chloroplast and the mitochondria. Okay? For us, we only have the mitochondria. Right? So the difference between us and them is they make their own food and then they burn the food. We have to eat the food and then we burn the food for fuel, for energy. Okay? So it's important to understand this balance, right? Any questions? I know it's really draining to have a lesson after the quiz, but try to stay with me, okay? Here we have another structure. This one is, again, only found in plants and not animal cells. Okay, I have a slide coming up that tells you what's plant exclusive and what's animal exclusive, okay? So like, you don't have to necessarily write down at this point. This whole thing is a plant cell, and a majority of the cell is taken up by this thing called the central vacuole. Okay, that is where they store water. Okay. Plants, they love to soak up water from the roots. And by storing the water in the vacuole, it allows them to use it later on, but it also allows the plant to kind of stand up against gravity. If you have any house plant and you forget to water them for a couple of days, sometimes you see the leaves kind of like wither and then it goes down, right? Yeah. But as soon as you water them again, they will stand up and prop open again, right? Storing the water in the central vacuole allow the pressure to build up in the cell, and that helps the leaves to open up. So generally speaking, they store water and nutrients, but sometimes they could store other things, like the colors for the flower. Of course, not the whole plant, but like only at the petals, cells of the petal. In some cases, they can also store poison. We're talking about like a poison ivy or something, right? All those poisonous compounds, they are stored in the central vacuole. Right? Once again, we do not have the central vacuole. Plants do. A central vacuole is not the only kind of vacuole that exists. We are back to this guy again. Bless you. This is a paramecium. Okay. And again, paramecium is not animal cell, it's not plant cell. They are called protists. And a lot of these protists, they live in freshwater environment. Ponds, rivers, whatnot. And one of the challenges when you're living in these open freshwater environment is that water keeps on coming into the cell. Right? And if the paramecium does not remove those incoming water, it will swell up like a balloon, and then it will pop and die. Right? So there is going to be a central vacuole here, which constantly squeeze and pump out the excess water to keep it from not dying. Okay, so we don't have these contractile vacuoles. Plants don't have them, but some other organism, like the protists, they do. Okay, so you'll see one here, you can see one. Another feature that a plant has would be the cell wall. The cell wall. So cell wall is found outside of a cell, and it doesn't regulate any movement. All things can pass through it pretty much. And as you learned before, it is made up of cellulose fibers, which is a kind of polysaccharide, right? You should know that since you just did your test. And their function is to prevent a cell from bursting. So for us, if you have too much water in your system, your cell will start bursting. Okay? Consuming too much water in a short period of time can actually be lethal. It's called water toxicity. But plants are okay. They have a cell wall. They love it when there's a lot of water. They are not going to burst. All right. Here is another concept check. Excuse me. Thank you. We're looking for the false statement. What's that? Try again, please. Oh, sorry. No. Yeah, you're right. Okay. There are more than one correct answer over here. Yeah, that's good. That's one of them. Yeah. No, yeah, that's one of them. Good. Abundance mean a lot, okay? Anyone else? Yeah. Okay. So let's see. We're looking for all the false one. Smooth ER is part of the endomembrane system. That's correct, okay? Endomembrane system includes the nuclear envelope, rough and smooth ER, Golgi, and lysosome. We have a list of it a couple slides ago. There is an abundance, a lot of mitochondria in muscle cells. Muscle, you move them all the time, right? So you need a lot of energy. That means you need a lot of mitochondria. So that's correct. We're looking for the false one. All cells have cell wall to protect itself from the environment. That's not true, right? We do not have cell wall. Hydrogen peroxides are found in lysosome. That's not true. Okay. Hydrogen peroxides are found in peroxisome, the first thing we learned today. Okay, peroxisome. Chlorophylls are found in chloroplasts. That's correct. Okay. These are the incorrect ones. All right, so that's what I mean, right? Like on your online quiz next Wednesday, there will be questions like these, right? Not a lot, like a couple of them. I don't remember how many. I'll tell you later. But you will have to choose all the correct answer. These ones are worth 2 marks each. 2 marks. Okay, you can get part marks. All right, so for every extra answer that you circle, you lose some mark. For every correct answer that you missed, you will also lose some marks, but you cannot lose more than 2 marks. Okay, so the maximum you can get is 2, the minimum you can get is 0. Okay. All right. I will skip these slides. I will not test you on it. We just simply don't have time. Okay. If you sign up the course to learn about cytoskeleton, I'm sorry, okay? Maybe you have to ask for a refund. So we're not gonna do this. Okay, we're not gonna do this. Not gonna do that. Okay, not gonna do that either. I will have to talk a little bit about my microtubules though, okay? Because they are kind of important. Okay, so this 1, 2, 3, 4. Okay, cross them out. We will not be learning them, okay? We'll do microtubules though, because it's kind of important, okay? Now, imagine the rough ER. Right? The rough ER makes a protein and it packages it in a vesicle, okay? Right? Don't confuse the vesicle with vacuole. Vacuole is like that big thing, right, inside the plant cell, or those like pumping vacuoles. Vesicle is what you store protein in, okay? So I want to ask again, where does that vesicle go? From the rough ER, where will it go next? Yeah, to the Golgi body, right? The Golgi body. But here is the thing. How does it know where to go to get to the Golgi. Okay, it's not like they think, right? Because cell is the smallest alive unit. Anything inside is not really alive, right? If we rely on random motion, then it might take a very long time for the vesicle to get to the Golgi. Okay, and then your cell is not very efficient. So it turns out inside your cell, there are these microtubules. Okay? This is a really short one, but typically inside the cell, if this is your cell, and then this is your rough ER, you don't really have to draw this, okay? But this is rough ER, and then this is the Golgi. There are actually tracks that is like all over the place inside the cell. You don't have to draw this really, okay? So the tracks are like conveyor belts, if you would. When a vesicle gets off from the rough ER, they will hop onto one of these microtubules, these conveyor belts, and then this is for real, okay? You can search it online if you don't believe me. There is a molecule called kinesin, which is like a person without a head, okay? And then it has 2 arms and 2 legs. It will grab onto the vesicles with the 2 arms, and then it will walk along the microtubules and carry it to where it needs to go. Okay, so if it needs to go to the Golgi body, it will carry it along the microtubule and drop it off there. Okay. Same thing, after you repackage the protein from the Golgi body, if it needs to be sent out of the cell, then it needs to hop on another microtubule, and then it will transport it all the way to the surface. So this reinforces the idea that nothing is random when it comes to living things. Okay. Everything has order to it, and there are complexity associated with it, even all the way down to the cellular level. Okay, so that's one function of the microtubules, to provide a track for things to move along. The microtubules can also provide structural support. Okay, as you saw earlier, different cells have different shapes, right? And in order to maintain those kind of shapes, you need to have the microtubules to kind of prop them up and to provide scaffold to keep them in a particular shape. Okay, another organelle I want you to know is the centrioles. Okay. Animals have them, plant cells don't. They look like those spaghettis that you can buy, the dry ones, and usually there is going to be a pair of them in the cell. So the centrioles, they will produce a special kind of microtubules called the spindle fibers. We'll learn more about the spindle fibers later on in lecture 7, but for now, these are the fibers that will guide the movement of chromosomes when the cell divides. All right, so just to go back and think about one of the 4 principles of the cell theory is that all cells comes from the division of pre-existing cells, right? So we have 46 chromosomes. When the cell divides, we have to make sure each of the new cell will get 46 chromosomes. Right? So like you get 46, you get 46, which means we actually have to move the chromosomes around. We have to move the DNA around. So to help guide the movement of the chromosomes, we have these special kinds of microtubules called spindle fibers. So we'll see how they work exactly later on. Finally, the microtubules also make up 2 other things. One of them is called flagellum. Flagellum is singular, and then more than one is called flagella. In humans, in animals in general, actually, in animals, the only place where you will find flagellum is on the sperm. Okay, so the tail of the sperm is a flagellum. There should only be one tail. Right? Sometimes it's possible for a sperm to have 2 tails, but it doesn't know how to swim. Like it just goes around in a circle. You can watch that on YouTube if you want. The other thing that the microtubule can form are these things called cilia. Okay, cilia, usually there are multiple things on them. Right? It covers the entire cell. So things like the paramecium again that we talked about, paramecium, they are covered with cilia. And again, the reason why we're talking about these things is because they are made up of microtubules. Okay. So beating of the cilia is going to help the organism swim around. Okay. We also have cilia in our bodies. For example, we saw this earlier, right? In the lecture, not today, but like last time. I showed you a picture of this. It was a like a grayscale, and you guys told me it's in the airway. Remember? Okay, so in the airway, you have these cilia that helps you sweep up pollutants, maybe, right? Debris that you breathe in, and then we can cough them out, or we can, blow our nose and get rid of them, right? So that's what they look like close up. It's all in our airway. Right? Heavy smokers, they don't have it, so they have the smoker's cough, right? I told you. In the female's body, there is another spot that has cilia. Men don't have these, right? Anybody know what this part of the body might be? It has these, cilia. And anyone can guess what that might be? No? Okay. So this is the fallopian tube. Fallopian tube. Okay. Sometimes it's called the oviduct. That's where the egg is gonna be in. Okay. So the fallopian tube is connected to the ovary on one end, and then do you guys know what it's connected to the other on the other side? The egg is released from the ovary. Where does it go? Ultimately, you know? I heard you see it. Saw you say something. No? Okay, well, you should take anatomy next semester and then we'll all find out. It's going to go to the uterus, okay? That's where the baby grows. So connecting the uterus and the ovary is going to be the fallopian tube, okay? So the egg doesn't know where to go sometimes, which is what these cilia are for, okay? Like if the egg drift up too much and then it goes to the edge, the cilia will be like sweeping. It's like, keep going, keep moving. You're not there yet, okay? So cilia in the airway and cilia in the fallopian tube. Both cilia and flagellum, they are made up of microtubules. All right. This is a slide that tells you the difference between animal cells and plant cells. Some of the stuff we haven't talked about, okay? But nevertheless, it's a good summary. For example, like centriosome here, I should change this slide. For us, centriosome is same as centriole. Okay, it's just like the same thing. Doesn't really matter. We can change that. So I will post a video, like an animation that I found online today as well. And you can watch that as a summary. Okay, it's a good summary. It basically talks about everything that we learned in the lesson. All right. So today's announcement is very important. Make sure you read it. Okay, it's going to be very long. There's a lot of stuff about what's coming up, and review videos and all that stuff. Okay. Yeah, we don't have to do this because we didn't learn all of the microtubules, all of the cytoskeleton, and that's it for lecture 3. Okay, but don't go yet because we have to start a little bit of lecture 4. But don't worry, I'm not gonna be talking to you again too much. We're gonna draw something. Once we draw it, and then we can go. Okay, sorry. We're almost there. We're almost there. That's why I don't like classes on Mondays, because every year, classes on Monday, it's like so rushed uh, because of all the holidays. Okay, lecture 4. Lecture 4 is called transport. We are going to be learning about how things get into and out of the cell. But before we understand that, we have to learn more about the cell membrane structure. Okay, so I'm going to draw it out for you. This is called the fluid mosaic. Mosaic, sorry, model. of the cell membrane. Okay, it's not a huge drawing. If you have a piece of paper, maybe we're gonna draw something here, here, and then we're gonna take some note down here, here. Okay, drawing notes. Okay? It's not gonna be too long. Okay, 15 minutes tops. So, fluid. The model, it's called fluid mosaic. Fluid means like it's kind of liquidy, right? It's not like the wall. We do not want a cell membrane that's like the wall. If it's like the wall, it's very rigid, it's solid, nothing goes through, and that's not what we need. Okay, we need to be able to bring nutrients in. We need to be able to send waste out of the cell. So that's why it is going to be fluid. It's dynamic. It's going to be moving around, okay? Mosaic. For those of you who don't know what mosaic means, let me just search it up for you so that you can see it. Mosaic. Window. Okay, that's a mosaic window. Okay, it's one of those windows where, you know, it's made up of many different pieces, okay? And then it forms a whole. So that's what the cell membrane is going to look like. Okay, it's not just one thing throughout and all looks the same. In fact, it's multiple different components kind of, you know, mix and match and connected together. Okay, so we're going to need Google image in a bit again. But for now, let's draw it out. So here we have a cell. I'm just going to do an animal cell. It doesn't really matter. And of course, that's the nucleus. And we are going to be taking a slice of the cell membrane. Okay, help me out here. What's the major component of the cell membrane? Yes, I hear someone say phospholipids, right? So we're going to draw a bunch of phospholipids, okay? Now, some of them are going to have saturated fatty acids. And every now and then you can pop in an unsaturated fatty acid. Okay. So just draw some of these things. Now leave a gap though, and then we're going to put more. And then I ask that you do it one more time, put another gap. And then we will have, that's pretty much it, okay, for now. And so I'll give you a moment to draw that. So this is called a phospholipid bilayer. Now, don't draw this next part, but I want you to watch. We are looking at like a 2D illustration of this, but you got to remember the cell membrane is actually 3D. It goes around the entire cell, right? So it looks more like this. Don't draw this next part. Just watch. Okay, so you will have like more heads, right? Do you understand? Like it spreads the entire one, right? So it's like the ball pits. You guys know ball pits? Like some people don't, I guess, like, you know, but I'll search it up for you. Kids ball pits. You know, like these ball pits, okay? What's going on? See result closer to you, nope. Okay? So if you think of a ball pit, right? When a kid goes into the ball pit, The balls can move around, right? Okay, that's exactly what's going to happen with the phospholipid bilayer. It's fluid. Okay, the phospholipids are able to, they are able to move sideways. They can go, kind of, in front, behind. That's normal. But they do not usually flip to the other side. Okay, so moving side to side within the same leaf is okay. Flipping is rare. Okay? I'll write that out for you. Individual phospholipid and move freely. Within one layer. They rarely flip to the other layer. Okay, understanding the structure of the cell membrane will help you answer a few questions on the lab. Right? So if you're stuck, make sure you come back to here to try to, find the answer. Okay, so of course, this would be like the outside of the cell. And then this would be the inside. This would be the cytoplasm. And this would be a hydrophilic head, which you already know. That as well, hydrophilic head. And then in between, that's where you will find the hydrophobic tails, right? Okay. Hydrophilic head, and then hydrophobic tails. And judging from the drawings, the tail portion, the hydrophobic layer, is much thicker than the hydrophilic heads. Okay? And that's going to have some implication when we talk about it again on Wednesday. Okay. Phospholipid is the major component, but it's not the only component. It's mosaic, right? So there are other stuff as well. And one of the other stuff that I want you to draw is there are going to be proteins here. That's one protein. That's another protein. Okay. And you might have a protein that's just going to be stuck on the inside or stuck on the outside. Proteins that goes all the way through, we call them integral. Integral protein. It spans the entire membrane. It goes all the way through. And you can see it on both sides of the membrane. It's like a raisin bread, okay? You can see the raisin on both sides. Sometimes the protein might just be stuck on one side. And if it's stuck on one side, we call it a peripheral protein. Now make it bigger. Also, lots of seats at the front. If you can't see at the back, next time you can sit closer to the front. Okay, what's another lipid that you will find in the cell membrane? Steroids. That's right. Specifically, what kind of steroid? That's right, cholesterol. Okay, so I'm going to draw some cholesterol here. You know, the 4 carbon rings. Hopefully you got that question right on the quiz. And so these are cholesterol. There are not a lot of them, just some of them. And the function of the cholesterols in the cell membrane is that they help control membrane fluidity. It helps control membrane fluidity. Generally speaking, more cholesterol equals to more fluid. Less cholesterol equals more solid. So it really depends on what kind of cell membrane you are looking at. Okay. For example, if the cell belongs to an organism that lives in a very hot environment, okay, very hot environment, you actually want to have less cholesterol in the membrane. Okay? Because if it's very hot, things tend to melt, right? Things tend to become more liquid. Right? And by making it more solid, you kind of counteract the high temperature. Similarly, if you are talking about a membrane from like a plant that grows in a very cold environment, you would want it to have more cholesterol. Because if it's very cold, then it tends to freeze up and become more solid. We don't want it to become more solid because then nothing can go through. So those plants, their cell membrane would have more cholesterol. Okay? We'll talk more about that next class. Just 2 more things, and then we're done. I promise. So we have the phospholipids, which are in black. We have the purple proteins, and then we have the orange cholesterol. Now, every now and then, some of the proteins, not all of them, some of the proteins, they are going to have some sugar on them. Right? These are like glucose. So that's carbohydrates. And some of the lipids might have carbohydrates on them as well. Right? So remember, when we say the Golgi body will modify the proteins, one such modification is to put carbohydrates on the protein, then send them off to the membrane. So this whole thing that has a sugar stuck on the protein, it is called a glycoprotein. Glyco means sugar. And you can probably guess this guy here with the sugar stuck on the lipid, it is called a glycolipid. Right, what do they do? I'll just quickly write a note here. They are important for cell recognition. Right? Cell recognition. Like, how does the cell know? Oh, I just realized the color is very big. It looks much darker on my screen. Let me see if I can change it for you. Sorry. Okay. So how does your body, like your white blood cells, for example, which defend your body against bacteria, how does how do they know what kind of cells belong to the body and what kind of cells belong to a bacteria? They need to be able to tell the difference, right? Otherwise, they would maybe attack your own cells or they would fail to attack the incoming bacteria. So it turns out on the surface of all cells, your cells, bacterial cells, right? All kind of cells, they have these glycoproteins and glycolipids. That kind of creates an ID tag, right? For the cells to kind of scan. So when your white cells see some ID tags that does not belong to the body, it knows to attack it. Okay. No two individuals are going to have the exact same ID tags unless you have an identical twin. Okay. Which is why you cannot just take an organ and then transplant it to anyone else, right? You have to match it. And even with the best effort to match it, it's not going to be 100% match, which is why your body could reject it. Okay. So how does it know? Well, it had to check on these glycoproteins and glycolipids. Right. Okay, thank you so much for your attention. I will see you guys on Wednesday.